begins the new session. So welcome to the hybrid and mixed session B and C. My name is Hélène Valentine. I work in Rennes at INRA in France, and my co-chair is Christian Botta, and he will introduce himself.
Okay, thanks, Hélène, for kindly introducing me. So I'm going to read the long title of my presentation, Towards New Strategies for Extending the Ground Beef Shell Life, the cases of electrolyzed water, bioprotective cultures, and metataxonomy-based surveillance. First of all, I want to say thanks to the committee that gave me this possibility to present here. I'm really happy to be again in presence, face-to-face, especially here in Athens. So let's move to the case of study. If you may understood, we are going to talk about ground beef. Why ground beef? Because it's, let's say, the worst-case scenario in terms of microbial contamination and perishability of the final product. So from the early stages of the slaughtering processing, from the carcasses are contaminated from the hides, especially if they are themselves contaminated from fecal residues. Along the processing line, this initial contamination grow step by steps, collecting spoilage bacteria from operators, the hands of the operators, saliva, but also cross-contamination between batches that is carried out by water and surfaces, and also gears, knives. So at the end, we reach the point of trimming, the mincing, the grinding passage, in which this contaminating, this spoilage microbiota is spread in all the products. And so we have a product in which the only hurdles to increase the shell life are the low temperature and the type of packaging. And this shell life is, let's say, proportional to the amount of bacteria or spoilage bacteria that have been collected during the process. So practically, the aims of this study, there are more than one study, is overall to find the most proper way to extend the ground beef shell life, applying different approaches. First, we tried with decontamination of the surface meat trimmings with electrolyzed water. We checked during the shell life the output of this treatment, performing plate counts, metataxonomic analysis, and combining these results with the volatilome, and also checking at the end of the shell life the potential presence of biogenic amines. In another study, we tried to exploit the self-protection capability of the ground beef by isolating or trying to isolate bacteria able to produce bacteriocins from the early stages of shell life, and then use it as a starters in this product that is not a fermented product against the contaminant bacteria, against the spoilage microbiota. So introducing them again in the final product. Going to the results, the first case of study, we treated trimmings for 90 seconds by immersion in electrolyzed water for at 100 ppm of free chloride. We decide this time and concentration in relation to previous laboratory experiments. We performed all this in collaboration with a company, 46 samples, 2 different production in 2 different periods were followed. So with treated and untreated thesis. Unfortunately, we cannot see the taxa. We have a problem with the taxa, but about the results, we observed an effective decontamination on the surface of the trimmings, but this was not anymore observable in the ground product, in the ground beef. On the other hand, we could observe a significant separation between the microbiological dynamics of production A and production B. So higher presence of lactic acid bacteria and total viable counts for the production A that showed overall, let's say, a faster spoilage tendency that was associated in terms of metataxonomic analysis with a higher biodiversity in the first 6 days. And also the presence, you cannot see, but I'm going to tell you by memory, higher presence of lactic acid bacteria like Streptococcus in the first stages, the yellow one. In both production, we identified common indicators of shelf life end in the Photobacterium phosphorum and Lactococcus species that were strongly associated with the production of biogenic amines, tryptamine and tyramine, and also negatively correlated to the linked amino acids. So we were able to discriminate between production in different periods. We were wondering at this point if we could discriminate between production, in the same production between batches of different origin. So different carcasses, but processed in the same environment, in the same condition, and with the same materials. From the microbiological point of view, from the counts, we could not discriminate the two batches. We could also, from the volatilomic profiles, is not visible, is not significant a segregation between the two batches, but is significant, especially for the, in this case, we analyzed also the microbiota, the fungal community. This differentiation between the two batches produced in the same day is possible with was possible with the metataxonomic analysis. Going more in deep, we can see that we have an initial transient microbiota that is common between the two batches, and mainly harboring Cocuria, Staphylococcus, and other bacteria collected probably from the skin of the operators and animals. But then the two, sorry, I have a problem with the pointer. The two microbiota are splitting, so we have a higher presence of canobacterial divergence and higher production as a consequence of acetoin and hexanal, so hop flower related, volatile compounds related to hop flower. And so we can see more clearly, I hope, here with this network analysis, in which we analyzed the co-occurrence between all the taxa, and we observed that we have a purple group of highly co-occurring taxa, so ASV that are going hand by hand in the data set, and so they are present all together. This purple group here is here represented with this purple triangle, represent the initial microbiota. Then we have a turnover, but it's different if we talk about bacteria between the batch A and B. In the A, we have the development of Leuconostoid gelidum. In the batch B, we have higher presence of Canobacterium divergensis, as I said, and also Lactococcus species. That, as we said before, is an indicator of shelf life end. So this is the signature of the original carcass, the signature of the original trimmings, while the purple module, these species, are the shared community that came with the grinding passage. At this point, since we state that we observed this difference, clear difference between the beginning and the end of the shell life, we try to exploit the potential self-protective effect of this product, trying to isolate from the early stages, from the day of production, bacteria able to inhibit what they are the spoilage taxa, so the consortia of bacteria that are present at the end, so in the spoilage product. We perform this by challenge tests with the agar well diffusion assay and other spot on low, searching for, also here is missing the color, searching for halos of inhibition. As indicator, we use as the first step the spoilage consortia collected from the same product. If we go on, we made for more than 700 challenge tests. We find out that 41 isolates from the time zero were able to inhibit this spoilage consortia. We tested all these 41 against several pathogens, and we found out that like 2 of them were able to inhibit Listeria monocytogenes. That is quite a big problem in the ground beef. So we proceed with these 2 strains, and for one of them, we confirmed the capability to produce an antimicrobial peptide, or more than one antimicrobial peptides, probably for sure nisin, and we're going to see later, but also something else because was able to resist in the agar well diffusion assay to the treatment with proteinase K. So this good guy is Lactococcus lactis, a species Ornie. So from the same genus, we have an endpoint indicator for the shell life, Lactococcus species, but at the same time, we can find also potential protective strains. We characterize this strain in terms of phenotype. It's able to grow low temperature, presence of salt, no so many resistance against antibiotics, and mainly is able to inhibit in liquid media, BHI, broad heart infusion, the growth of Listeria monocytogenes. At the same time, looking inside the genomes, we observed the full operon, the whole operon for nisin, but also other gene codifying. We are trying to understand the mechanism behind, but this is another story that I hope to tell you in the future. Right now, we tried to apply what is this promising strain in several replicates in the same product, so in ground beef, but unfortunately, in this case, we couldn't observe the same inhibitory capability as we saw in the synthetic media. This is probably because here we are in a solid phase, not liquid. There is no direct contact, or it's difficult the direct contact between Listeria and these bacteriocin-producing strains. Going at this point to the conclusion, making a, let's say, a connection between these that are different studies, we can say that the treatment with electrolyzed water is unlikely to be an effective strategy to ameliorate, to improve the quality if the product is already in a good condition. Of course, this doesn't mean that electrolyzed water is not working against high concentration of pathogens, but this was not the case of our study. Ground beef shell life depends, we understood, on the pre-grinding microbiota composition. We observed also signature of perishability, signature of shell life, and the presence, for example, of Lactococcus species. This is quite interesting because it means that we can understand probably the future of a batch of ground beef in relation to the initial microbiota composition of the trimmings or the carcasses. About the bioprotection, we found there is this possibility we isolated in early stages bacteriocin-produced strains, able to grow in this at low temperature, but it often has happened in this kind of study. In vitro results are not always confirmed, as in this case, by the application in vivo. So the best, if we want to have a takeaway message, the best possibility could be to create a benchmark database of what is a good and bad metataxonomic profile. And in relation to this, and together with the initial counts, bacterial counts, define a batch-specific shell life. This could be also a way to finally move the metataxonomics, the more user-friendly techniques of the omics family, from the academic research to the laboratory of companies. I want to say thanks to all the co-authors, and also thanks especially to the students involved in the study, to the several companies, and all the others collaborators. And of course, thanks to all of you for your attention. If you have any question, please raise your hand.
Thank you, Christian. We have time for one question.
It's a bit long.
Nobody? Yes, here. Please.
Yeah, thank you very much for your presentation. I have a question on the culture that you tried to use in beef, and it didn't work. Were you sure that the nisin or the compounds that was produced in vitro was produced also in ground beef?
Thank you. That's a good question, and this is why we are trying to figure out right now. When I say I hope to tell you also another story, I mean how this Lactococcus lactis horniae is producing. If it's releasing of its, we are thinking in the last experiment we observed that maybe it's something bound to the membrane of the wall or the wall of the bacteria. So this is probably a reason why when we are in a liquid phase, it can reduce it can limit the growth, not limit the growth of Listeria, but when we are in a solid matrix like the ground beef, is not effective. We have to understand first the mechanism, and then try if we can figure out another application. Maybe on not on ground beef, or we can use on stick on the surface. This could it would help to have a contact between pathogen and the bioprotective strain.
So I have just a question for you. Did you check that there is no problem with the volatile compounds produced by Lactococcus?
We didn't check with the GCMS, so chemical. We perform anyway organoleptic. I didn't have here this information. We performed in parallel with the, of course, not in the sample inoculated with Listeria, only the one inoculated with the Lactococcus and the one control. We performed a sensory analysis, and a panel of more than 100 people couldn't recognize by odor, in this case, a difference. Of course, if the idea is to implement this in a real product, we have to pay really the attention on what is produced after the 10th day, for example, in terms of volatile compounds.
Okay, thank you very much. So thank you for this clear presentation and for answer. We can pass to the next presentation. So I may inform you that the second presentation, initially dealing with E. coli, has been postponed to tomorrow morning. So now we are waiting for Maria Eleni Dimitra Kopoulou. She will speak about traditional Greek products, but I can't name it.
Αυγοτάραχο Μεσολογίου. That's it?
Yes, hello. Good evening. My presentation is about DNA-based tools for authentication of Greek PDO Αυγοτάραχο Μεσολογίου. A few things to start with. Food authentication is a process that validates label information about the food origin, geographical or genetic, and information about production procedures. In fact, it is also an essential requirement for food quality. Why do we choose DNA-based tools? At first, DNA is an extremely stable and long-lived biological molecule that can be recovered from a biological material that has been under stress conditions. And then DNA can provide us more information than proteins due to the degeneracy of the genetic code and the presence of large non-coded stretches. So, DNA-based methods could contribute against food frauds because of their advantages such as simplicity, accuracy, and robustness. The aim of our study was to explore whether unique biological barcodes or markers of a high valuable Greek PDO product could be discovered. What is αυγοτάραχο μεσολογίου? The eggs from fishes, Mugilcephalus, caught in the Mesologi lagoon, are known with the trade name αυγοτάραχο μεσολογίου. The raw material of the product is derived from flathead mullet caught in Mesologi. Production, process, and preparation of this Greek PDO product are completed at the same region, and this product has a great commercial value, over 100 euros per kilogram, and constitutes one of the oldest in the category of fresh fish. Our steps of experimental procedure. At first, we started with a sample collection, followed by DNA extraction, selection of gene target, PCR amplification, and finally, we choose 3 molecular techniques, PCR-APD, PCR-FLP, and PCR-DGG. So, this is the map. In red highlight, we can see the geographical regions where the samples were collected, Greek PDO from Mesologi, samples from Mauritania and from Australia. Protocol for PCR-APD analysis. At first, we performed Enterobacteriaceae analysis with microbial cultures in VRBG medium. We extracted DNA from cultures and we proceeded with PCR amplification using random 10-mer primers, M13, 1245, 1290, OPA10, and OPA15. For PCR-FLP method, we extracted genomic DNA of samples. We amplify our DNA targeted to COI gene. Our applicons were 655 base pairs, and we used primers FIS F1 and FIS R1. Then we sequenced these applicons from COI gene, and finally we selected appropriate restriction enzymes. Protocol for PCR-DGG was started by bacterial DNA isolation as well. We amplify DNA targeted to 16S rRNA gene. A more specific V3 variable region of bacterial 16S rRNA from raw was amplified using primers GC338F and 518R. PCR products were analyzed by agarose gel electrophoresis and then were analyzed by gradient gel polyacrylamide. Recently, emulsion PCR-DGG has been proposed as an alternative tool for microbial analysis. Through emulsion PCR-DGG, we can obviate and face up limitations regarding conventional PCR. Emulsion PCR is implemented in third-generation PCR system known as droplet digital PCR. By means of emulsion PCR, we can produce droplets of DNA molecules in thermostable water in oil emulsion. And finally, genetic material is divided in droplets which contain more than one gene and is amplified in millions of independent reactions simultaneously. So, results of PCR-APD analysis in this figure, we can see cluster analysis of 16 sRNA binding profiles of bacterial communities extracted from fish eggs from different geographical regions. At 52%, we achieve total discrimination among samples. Figure 2 shows multiple alignment of the sequence of COI gene obtained from fish from different geographical origins. In green highlight, we can see genetic variations between samples. And finally, in table 1, we can see predicted restriction sites of 4 enzymes, HIN, FALU, PILE, and IQ3, and the generated pattern from PCR-RFLP analysis. As we can see, by using IQ3 enzyme, PCR product from mesology PDO was not digested. So we have also here total discrimination among samples from different geographical origins. Results from PCR-DGG analysis. We have two different electrophoresis conditions. In figure A, we ran electrophoresis gel 16 hours at 65 volt, while in figure B, we ran electrophoresis 5.5 hours at 190 volt. With red highlight is Greek PDO of Gotaro Mesologiou, with yellow samples from Mauritania, and with blue samples from Australia. Results of emulsion PCR DGG. In this figure, we can see a comparison between emulsion PCR DGG and conventional PCR DGG analysis. By emulsion PCR, we achieved to obtain 3 additional bands, so we can assume that emulsion PCR is more accurate and sensitive regarding bacterial communities, analysis of bacterial communities. So, in general, DNA-based techniques can provide a biological marker for any food type. They can also be effective by analyzing fungi or yeast or other genes of a foodstuff. And finally, combination of these methodologies could be unique biological barcode and which will be accurate and valid. We performed 3 different molecular DNA-based techniques in order to explore whether a unique biological barcode could be discovered based on either genomic DNA or DNA from bacterial communities. Band profiles resulted in these molecular techniques could be used as a barcode to certify the origin and authenticity of a Greek PDO of Gotara Mesologiou. And it is worth mentioning that it is the first study that is successfully applied DNA-based methods for authentication and traceability of Greek PDO of Gotara Mesologiou. Finally, considering the results of the study, adoption of DNA-based tools seems to be very promising for determination of food geographical origin and authentication among different samples. In terms of global food trade, implementation of these techniques in food industry could provide a great impact regarding quality SIMs, food labeling, food safety, and food fraud incidents. And in conclusion, further DNA analysis and application to a variety of foodstuff in a large scale may be the proper solution to manage adulterations and to strengthen consumer confidence as well. Thank you very much. Thank you for your attention.
Thank you. So we have time for 2 questions.
Okay.
I have one question. Are you planning or thinking to move also other than DGE on other metataxonomic, for example, analysis metagenomics to characterize?
We are planning to
In relation to the
study
viability.
at first other genes, and then we are planning to go metagenomics.
And how can we eat this kind of Greek product?
So, I'm sorry, I don't
How
hear.
can we eat this kind of Greek product?
Of how, what, when?
It's not a scientific question, it's how to eat.
A culinary question.
I think raw with spaghetti.
With spaghetti.
Okay.
Thank
Thank you very much.
Okay, I'm gonna announce you the next speaker, Samantha Dutoit, with a presentation entitled Whole Genome Sequencing and PCR RFP Characterization of Listeria monocytogenes, Isolates from Ready-to-Eat, Hummus, Fresh Produce, and the Food Processing Environment in South Africa. The floor is yours. You have 13 minutes.
Thanks. As announced, my name is Samantha Dutoit. I am a MSc Food Science student at the University of Stellenbosch in South Africa, and it's such a privilege to share part of my research project with you this evening. In short, the surveillance study contributes to the knowledge and understanding of the genetic diversity of Listeria monocytogenes in ready-to-eat hummus, fresh produce, and the food processing environment in the Western Cape, South Africa for the years 2018 to 2021. For ease of speech, Listeria monocytogenes will be referred to as L-mono here onwards. The main objectives of this study were to lineage type 60 L-mono isolates using polymerase chain reaction restriction fragment length polymorphism, and to identify the sequence type and serotype of a subset of isolates using whole genome sequencing and bioinformatic analysis. As you may know, L-mono is ubiquitous in agricultural environments and persistent in food processing facilities. Consequently, a variety of foods are contaminated and become serious health risks to immunocompromised consumers. The listeriosis outbreak experienced in South Africa a few years ago was evidence of this. The pathogen causes flu-like symptoms in healthy individuals and life-threatening invasive disease in vulnerable groups. Although the incidence of listeriosis is usually low compared to other foodborne diseases, most cases require hospitalization, and the disease outcome is often more serious. Infection is almost always caused by the consumption of contaminated foods, including ready-to-eat hummus and fresh produce. Both categories have been linked to listeriosis outbreaks and sporadic cases in the past. These foods are particularly concerning as they are typically stored at refrigeration temperatures for long periods of time and are consumed with minimal processing or no heat-at-home step. Collecting surveillance data for foods less studied supports a precautionary approach to food safety. Lack of awareness could have severe consequences to public health. L-mono can be classified below the species level into 4 lineage groups and 13 common serotypes. These serotypes can further be divided into sequence types. Only the sequence types of relevance in this study are presented on this slide. Different subtypes of the bacteria are associated with varying degrees of virulence and different clinical outcomes. Lineage 1 and serotypes half A, half B, and 4B are most commonly linked to human listeriosis and are therefore considered most pathogenic. Recorded trends allow the lineage types, serotypes, and sequence types of isolates to predict their potential to cause disease. Subtyping of L-mono from foods provides some insight into the potential risk these products pose to immunocompromised consumers. All samples included in this study were received from an accredited food laboratory called Microchem. A sample set of 60 isolates was selected from many received over the years. Samples were grouped into 3 categories. Ready-to-eat hummus samples ranged from an unspecified or original variety to jalapeno, za'atar, peri-peri, orange, red pepper, and coriander chili. Fresh produce samples included potato, cucumber, coriander, cling peach, leeks, spinach, and unspecified vegetables pre-wash. Food processing environment isolates were obtained from a variety of different facilities. A few environmental origins included floors, drains, food contact surfaces, equipment, and cleaning tools. No duplicates were added to the sample set from any category. All isolates were received on rapid almonochromogenic agar. To ensure isolate viability, presumptive positive almono was restreaked onto rapid almono plates. Colonies exhibiting characteristic almono growth were then streaked onto 2% blood plates for enrichment. DNA extraction was performed using a quick DNA fungal bacterial mini-prep kit. PCR amplification of the HLY gene genotypically confirmed isolates to be almono. Lineage typing was performed using the RFLP method developed by Rip and Goos. Isolates were essentially classified into one of 3 lineage groups based on shared single nucleotide polymorphisms. The HLY amplicons were subjected to restriction digestion using 3 enzymes. Different enzymes cut at certain positions specific to different lineage groups, producing fragments of varying lengths. A subset of 20 isolates was selected for whole genome sequencing. The subset comprised of 10 lineage 1 isolates and 10 lineage 2 isolates from a range of years. The purity of all DNA extracts was measured before being sent for whole genome sequencing. Whole genome sequencing was performed by 2 service providers, namely Cosmos ID and SAF, using an Illumina platform and an Ion Torrent platform, respectively. In silico sequence typing involves submitting the raw reads for the 20 isolates to the MIST 2.0 tool from the Center for Genomic Epidemiology in FASTQ format. Serotypes were then inferred from this data. Binomial and chi-square tests for equal proportions and chi-square and Fisher exact tests for independence were performed to identify significant associations between categories and isolate typing results. A p-value of less than 0.05 was considered a statistically significant difference. PCR results confirmed all presumptive positive isolates to be almono. Of the 60 isolates lineage typed, 57% were found to be lineage 1 and 43% to be lineage 2. There is cause for concern considering the pathogenic potential of lineage group 1 and its overrepresentation in the South African food industry. Other South African studies have come to similar conclusions. The 20 isolates sequenced were found to be of 7 sequence types belonging to 3 serotypes. The most common sequence types were ST5, ST121, and ST204. No novel sequence types were recognized from this research. Serotypes half A, half B, and 4B are collectively responsible for 95% of all described human listeriosis cases. All fresh produce-related outbreaks listed previously were caused by one of these 3 serotypes. The serotype 4B ST6 strain, responsible for the 2017 to 2018 listeriosis outbreak in South Africa, was not identified in this study. However, clinical isolates analyzed during the time of the outbreak were represented by 9 sequence types, including ST1, ST2, ST5, and ST101. A South African study by Mafuna et al. Suggested ST1, ST2, and ST204 to be particularly virulent due to the presence of many main virulence factors. At an alpha level of 5%, samples for the food processing environment and ready-to-eat hummus did not provide sufficient evidence to conclude that the proportions of lineage types were significantly different. However, lineage 1 was significantly associated with fresh produce. For this category, all isolates but 1 were found to be lineage 1. Similar South African studies have also reported a prevalence of lineage 1 in fresh produce. Kyoda and Okey recently characterized all 90 Almono isolates analyzed from fruits and vegetables in the Eastern Cape province as lineage 1. Both this finding and the results obtained in our study should not be overlooked, especially considering the ready-to-eat nature of many of these foods. Both serotypes half A and half B were recovered from all 3 categories, while serotype 4B was only found in fresh produce. No single sequenced serotype was significantly overrepresented in any of the categories. Serotype 4B, isolated from potato and leeks in different facilities, is considered the most pathogenic serotype. This serotype has previously been recovered from various fruits and vegetables in many different countries and has caused listeriosis outbreaks associated with ready-to-eat salads, caramel apples, stone fruit, bean sprouts, and leafy greens in the last 10 years. No found studies have identified the sequence type or serotype of isolates from ready-to-eat hummus. However, one publication deduced a strong association between infection, particularly by serotypes half A and 4B, and the consumption of hummus prepared in a commercial establishment in America. ST204 was most prevalent in the food processing environment, ST5 in fresh produce, and ST5, ST101, and ST121 in ready-to-eat hummus. Again, no significant associations between sequence types and categories were identified. Mafuna et al. Identified various stress tolerance genes, benzalkonium chloride resistance genes, and biofilm formation genes in ST204, and reported this sequence type as well as others to be well adapted to the food processing environment. Another study by Naditz et al. Found ST5 and ST121 to contain plasmids contributing to food processing environment-associated stress survival. ST3 was only recovered from the food processing environment. ST1 and ST2, both serotype 4B, were only found in fresh produce, and ST101 was only sampled from ready-to-eat hummus in this study. Factory origin information of the isolates was requested from the supplier after analysis for discussion purposes. The contamination of ready-to-eat hummus either originates from raw ingredients or, more commonly, from cross-contamination within the food processing environment. It was noted that the same ST5 serotype half B strain was recovered from a hummus blender and a ready-to-eat and hummus blender and 2 hummus samples, original and coriander chili varieties, and coriander in the same facility, this bottom ready-to-eat and deli food factory, in 2019. This finding suggests possible cross-contamination within this facility and emphasizes the importance of good manufacturing practices, appropriate factory design and production flow, correct storage of raw ingredients and ready-to-eat products, and effective cleaning and sanitation. It was noted that 3 of the 4 factory origins supported the survival of more than one Almono strain. It was also evident that ST5, ST121, and ST204 were able to proliferate in very different processing environments. Relatedness analysis revealed distinct associations between lineage types, serotypes, and sequence types. Lineage groups and serotypes within lineage groups clustered separately. Isolates of the same sequence type clustered with 100% similarity. The 2 serotype 4B isolates clustered closely and were more strongly related to each other than to the other isolates. This sort of genetic profiling is very limited for Almono in South Africa, especially for the food groups of interest. Serotype and sequence type results for hummus constitute the first of their kind. Only one very recently published South African study has serotyped Almono from fresh produce in the Eastern Cape using an agglutination technique, and no found studies have identified the sequence type of isolates in this category in our setting. In summary, lineage 1 seems to persist in the South African food industry for reasons that still need to be explored in future studies. The continued recovery of lineage 1 from fresh produce suggests this lineage type to be well adapted to proliferate in these foods and potentially their cultivation environments. the significant association between lineage 1 and fresh produce, and the recovery of serotype 4b isolates from potato and leeks present this category as a higher risk. The recovery of serotypes commonly linked to human listeriosis from ready-to-eat hummus also suggests this product to be a potential health risk to pregnant women and immunocompromised consumers, especially considering the ready-to-eat nature of this food. Serotypes half A, half B, and 4B are evidently able to overcome the stresses implicated in the South African food industry and exhibit a competitive advantage over other serotypes. Sequence types detected in this study are reportedly prevalent in other parts of the world and have been responsible for listeriosis outbreaks, suggesting them to be a potential threat to the public health of South Africa. I would like to acknowledge the following people and organizations and their contributions to this project. Thank you for listening.
Thanks, Samantha, for your presentation. We have time for one question from the audience. If no, thanks again.
Thanks.
I'm glad to announce you the next speaker. That is my co-chairman, Dr. Hélène Valentin from HIRE, with a presentation entitled Metatranscriptomics and Metabolic Modeling to Identify Bacterial Metabolic Interaction During the Manufacture of a Modern Processed Cheese. The stage is yours.
Hello, everybody. So today I propose to explore the potential of metatranscriptomics and metabolic modeling to identify bacterial metabolic interactions during the manufacture of a model pressed cheese. So you know that the activity of cheese microbiota is responsible for most of organoleptic qualities of cheese. The cheese are characterized, as many other fermented foods, by successive growth of microbes, specific and complementary microbial metabolisms. So we could ask the question, what are the metabolic interactions between these microbes and what are their role in the functioning of the global ecosystem? So today our scientific questions will be, which species contribute to the acidification? Which species contributes to the aroma compound production? Which metabolic pathways are activated and when? And finally, which bacterial metabolic interaction contributes to the functioning of these microbial ecosystems? So our strategy was to sample cheese throughout making and ripening. First, we did numeration to assess culturable bacteria, and we also did a targeted metabolomic to quantify sugar, acid, and aroma compounds. Finally, we performed a metatranscriptomic analysis by sequencing RNA from bacterial cheese to give and to obtain a snapshot of metabolism of each of the species identified. So the cheeses were semi-hard cheeses, and we performed the manufacture in our laboratory in a pilot plant. The milk first was inoculated with 3 strains, Lactococcus lactis biovar diacetylactis, Lactiplantibacillus plantarum, and Propionibacterium fraudenreichi. The making was traditional. At the end, the cheeses were vacuum packaged to prevent yeast and fungi. And there was a 2-month ripening period. So we sampled the cheese throughout the making, and at all sampling steps, microbiology and biochemistry were performed. We also did molecular biology and metatranscriptomic only at 4 steps, at molding, demolding, and 2 of the ripening steps. Microbiology enables us to monitor growth of each species. Biochemistry and in particular chromatography gave quantification of sugar, acid, and aroma compounds. We also did complete DNA extraction to get a complete genome of each of the 3 species, and RNA extraction. So the DNA was sequenced by both parent Illumina and PacBio, and RNA was only sequenced by Illumina. The computational methods, so we did a genome assembly using Unicycler, an automatic annotation of the genome using microscope platform, and a reconstruction of metabolic pathways using the pathway tools. The metatranscriptomics was done by mapping all the reads obtained by Illumina on a genome sequence using bowtie, and the reads were counted on each genes of the genomes. A normalization step was performed, and all the results I will show you today is a set of differential genes identified using EdgeR, and all the results will be expressed as a logarithm of fold change. So we checked the repeatability of semi-hard cheeses before doing molecular biology, and the repeatability was very good in terms of humidity, fat, dry matter, and chloride, but also in terms of bacterial growth. Most of the examples I will show you today will be taken during these steps at acidification steps between molding and demolding. So the genomes obtained were complete with chromosomes and plasmids thanks to Unicycler assembly. And we did the mapping of RNA reads on these genomes, so we can observe all induced and repressed pathways throughout the making and the ripening, but I will choose for you some example of carbon metabolism involved in bacterial interactions. So first, the milk contains 2 sources of carbon. One is a citrate and the other one is a lactose. So first, the citrate was the metabolism of citrate was observed, and the citrate was rapidly utilized between molding and demolding, according to the previously described pathway. In lactis, the induction was a twofold. The same pathway was also induced in L. plantarum, and in this case, we can say that the metabolic interaction is a competition for the citrate. And we have a functional redundancy using the same pathway induced exactly at the same time in L. lactis and in L. plantarum. Secondly, the lactose was also utilized by L. plantarum, and for the majority between molding and demolding. So the lactose was split into glucose and galactose, which was then metabolized for lower pathways. All genes were induced 5 to 7-fold in L. plantarum and 15-fold in P. freudenreichi, and surprisingly, at these steps, it was stably expressed in L. lactis. However, in L. lactis, we observe another pathway which was scarcely described in literature and which was induced at the end of the ripening. It is the tagatose pathway involving a permease and a phosphorylation of lactose. All these genes were placed on arbor on plasmid. My last example will be the production of acetylene, a typical cheesy aroma compound. And we noticed production of acetoin just after the molding. The previously described pathways was induced, 2 to fold change for L-lactis and L-pentarome for the first steps in red, and was highly induced in P4.IC with 14-fold. In this case, for acetoin, we can imagine a division of labor if we think that the acetolactate can pass through the membrane and through the cell wall of lactic acid bacteria to fulfill the P4.IC. This interaction is characterized by a functional redundancy with specific induction of genes. So to resume just one slide, we get the successive carbon metabolism and the interaction. So first, the lactic acid bacteria produce lactate from lactose and citrate, and also acetoin. The lactate and the lactose was utilized in P4.IC to produce propionate and acetate. And at the end of the ripening, an acetoin production still arrived. So the bacteria seems to have a good team spirit for degradation and production, but one question remains: do they exchange additional metabolites, more than lactate and acetolactate? So we performed metabolic modeling to identify metabolic exchanges. So metabolic modeling was previously presented by a colleague from Israel, I think, today. So we converted the annotated genomes into genome-scale metabolic model, first at individual species for individual strains, and secondly for ecosystem, by mixing the three models. And under biomass constraints, we checked that flux were in agreement with metatranscriptomics and targeted metabolomics previously described. So we can show here a flux of lactose and also a production of lactate and acetolactate pathway. This metabolic model at ecosystem level enables us to detect a potential exchange. The exchange of lactate between lactic acid bacteria and propionic acid bacteria has been previously published, but we also identified an exchange of ribose, succinate, glycerol, and at the expense of both lactic acid bacteria, and also probably an exchange of serine and phenylalanine to the benefit of P4.IC. So we have to confirm, going back to wet lab. The take-home message today was metatranscriptomics enable us to identify induced pathway in each species, and it gives also a snapshot of successive metabolism during the cheese making and ripening. We also identified microbial competitions like citrate and lactose utilization, and division of labor for production of acetoin. The metabolic modeling gives us clues to metabolic exchange between the species. So the perspective, the strategy applied here can be applied to any fermented foods to decipher metabolisms and interaction. And since most identified pathways are species and strain dependent, this kind of knowledge could be very important to select and to combine strains to develop new fermented food products. So I will thank all my colleagues from INRA and INRIA, and in particular our engineer Wenfan Kao and my PhD student Maxime Lecomte. And thank you for your attention. Thank you.
Thank you, Hélène, for your really nice and detailed presentation. we have time for a couple of questions from the audience. If no question from the audience, I have one curiosity. No, not from the audience. Okay. I saw in one graph production of butanoate, if I catch it. Do you have any, idea if it can be produced from one of these three, or about the pathway that can be involved? If it's
Which metabolite?
butyric acid, butanoate.
Butanoate. We don't know exactly the pathway for this kind of product, probably because we have Propionibacterium fraudenreichi, and we know that this species is hardly involved in lipolysis in cheese, in pressed cheese. The butyrate probably comes from lipolysis of milk fat, but we are not sure.
Okay. Any question? No more? Okay, thanks again.
Okay, thank you.
Ms. Moine, thanks again. Sorry, but I don't have the agenda. Okay, so let's move on with the next presentation. Paulina Sredrinka with the presentation entitled Optimization, it's online, okay. Optimization of the in vitro model of gut microbiota culture and application to toxicomicrobiomics studies. Paulina, the stage is yours.
Okay, so to begin with, I will provide a short introduction about gut microbiota and its interactions with xenobiotics. So, microorganisms gastrointestinal tract constantly interact with each other and with the host, playing an important role in maintaining.
Sorry, we can't hear you again.
We still have problems, but I think it's not your fault.
Can you hear me?
Yes.
Yes, now, yeah. Let's try like this.
Okay. Microorganisms inhabiting gastrointestinal tract can interact with each other and also with the host, playing an important role in maintaining human health. So this symbiotic balance can be easily disturbed by numerous factors leading to dysbiosis, which may have adverse health effects. So currently there is a great need to optimize methods of in vitro culturing of human gut microbiota to investigate and understand its link with pathogenesis of various diseases, but also in the comprehensive evaluation of environmental pollutants toxicity. So a branch of toxicology that explores such xenobiotic microbiome interactions is called...
Sorry, I have to interrupt you again. I'm really sorry. Can you just move a little bit down the camera so that we can see also your face? Perfect. Okay, let's start again with the time. I'm sorry.
So can you hear me and see me?
Yes.
Okay.
Yes? Okay. So the branch of toxicology that explores xenobiotic microbiome interactions is called toxicomicrobiomics. I want to emphasize that one of the important roles of human exposure to xenobiotics is its migration into food from food contact materials. So in my research, I study mainly bisphenols, which are an example of xenobiotic migrating into food from food packaging. Xenobiotics ingested by the gastrointestinal tract have a significant impact on the intestinal microbiota. These compounds can be directly metabolized by microorganisms, which in turn may lead to the activation or biological inactivation of these compounds, as well as the production of toxic metabolites. Xenobiotics also indirectly influence the taxonomic structure and metabolic activity of intestinal microbiota. They can disturb the production of many signal metabolites, which are important for the host health. So the aim of my research was to optimize the methods of growing the gut microbiota so that the culturing process would allow us to maintain the high biodiversity and metabolic activity of the microbiota, and that it could be used in further studies evaluating impact of various xenobiotics. So gut microbiota samples were collected from 15 healthy donors, pulled together and frozen in a cryopreservation buffer. The anaerobic cultures were carried out using 4 different media selected from the literature. The incubation was for 24 hours in anaerobic workstation. The samples were analyzed for changes in taxonomic profiles using Illumina-MySeq platform. Short-chain fatty acid content was analyzed using gas chromatography, and analysis of the untargeted metabolomic profiles was conducted using liquid chromatography. The tested media were inoculated with pullet stool samples marked as mix and incubated in vitro batch model for 24 hours. Then post-culture samples were treated with PMA viability dye to remove non-viable bacteria DNA from sequencing and to reduce viability bias. Obtained outcomes were compared to the non-cultured mix inoculum, representing the initial structure of gut microbiota community. Here on the screen you see taxonomic composition of gut microbiota.
Sorry.
Presented as mean of 6 replicates.
Sorry, we can't hear you again.
Can you hear me now?
Stop.
Really low volume. I don't know if it's a technical problem here or the microphone from your side.
No.
Here it seems okay, so try to...
Can you hear me now?
No, it's not enough.
Not well. Audience cannot hear you, I think.
No, it's impossible.
I don't know if it can...
Is it now better?
Oh,
Yes,
okay, now.
it's better, yes. Sorry. What's that?
I'm sorry.
So you explain the mix of different medium.
Yes, here on the screen you can see taxonomic composition of gut microbiota presented as a mean of 6 replicates at phylum level. Only the relative abundance of Bacteroidetes and cyanobacteria was significantly different between media groups and non-cultured inoculum at phylum level. Next are 5 alpha diversity metrics were calculated. The non-cultured mixed inoculum was characterized by a high species richness and evenness by applied diversity measures, and after 24 hours of culture, no significant differences in the SM were observed in terms of CHOW1 index, observed features, pillow evenness, and fate PD index. However, in general, in vitro cultivation of gut microbiota was associated with depletion of the microbial community. In all of studied media groups, we noticed a significantly lower Shannon index, which is a quantitative metric of the species richness and the relative abundance. Next, we use PyCrust analysis based on 16S rDNA gene data, which predicted significant differences in the functional potential of the gut microbiota, non-cultured mix inoculum, and 4 media groups. The results of PyCrust predicted PyCrust prediction was an increase in relative abundance of genes associated with xenobiotic biodegradation and metabolism pathways, and it was observed in all media groups. Next step was evaluation of production of short-chain fatty acids by gut microbiota in 4 different media. Short-chain fatty acids are the end products of anaerobic microbial fiber fermentation, and they have been found to play important roles in various physiological processes, especially energy supplement, gut barrier integrity, and gut-brain axis. The highest total short-chain fatty acids production after culture was observed in SM, whereas the lowest production was observed in carbohydrate-free basal medium and fermentation medium groups. Next step was the evaluation of untargeted metabolomic profiles. A total of 37,000 compounds were detected in fecal samples after fermentation in 4 different media, whereas almost 4,500 compounds were significantly different between tested media. However, almost 80% of these metabolites were unidentified. So differences between gut microbiota metabolomes are shown on the left on the PCA plot. We see similarities between SM and FM groups, whereas
Sorry, Paulina, but the connection is very bad, and we can't hear you very well. So I think
maybe if you want to get to the conclusion, give the final message. If we can still hear you, because we are running out of time, and the connection is really going and coming back. And okay, let's try. I'm sorry.
So our results showed that of different cultivation media on global human gut microbiota community composition and in metabolomic activity. In our study, we observed that the use of low carbo media like a fermentation medium and led significant depletion of gut microbiota community and reduction of short-chain fatty acid production. Whereas Schedler medium marked as SM allow us to preserve the highest biodiversity and metabolic activity of microbiota. Moreover, we observed significant enrichment of the biodegradation pathways of xenobiotics in this medium.
Thank you, Paulina. Despite all the problems, we can understand the big amount of work, and it's a really nice presentation. Since we are running out of time.
yes, I think we have to keep the flow, and perhaps you can use the tools of chat you have on the app to connect people and to exchange with them. It will be easier, I think, by chatting.
Okay, thank you.
Okay, sorry about that. And thank you very much. So the next speaker is Meta. The next speaker is Meta Sterniza from a university of Slovenia, and she will speak about the Pseudomonas fragi biofilm.
Hello, thank you for the introduction. So my name is Meta Sterniza. I'm from the Biotechnical Faculty, University of Ljubljana, and today I will present a topic about Pseudomonas fragi biofilm and how it improves Campylobacter jejuni stress response in aerobic conditions. So first about Campylobacter. This is still the leading cause of human gastroenteritis reported by different organizations, and it's a zoonotic infection. Most of cases are sporadic, and they are connected to consumption of undercooked poultry meat or because of cross-contamination. And the outbreaks are mostly connected with unpasteurized cow's milk. But we can find Campylobacter also in water, and it was even reported in shellfish. What's characteristic of this bacterium that's on one side maybe making problems in the laboratory, but on the other side makes really big challenge for these bacteria in the environment is their microaerophilic and thermotolerant demands for growth. That's why these bacteria must overcome several stresses during their foodborne transmission. Now on the other side we have these really good adaptable bacteria which cause spoilage of food, and Pseudomonas fragi is one of the most commonly reported spoilage of Pseudomonas species, and it's psychotrophic. In recent years, there's been an increase in reporting that these bacteria can provide advantageous conditions for other bacteria. We were interested in the combination of these two bacteria that can be found in poultry meat, and we were interested at their growth at low temperatures. So our two first combinations were to test how these bacteria grow if they are co-cultivated, incubated at two different temperatures, 5 and 15 degrees, and we used two different growth media, so Miller-Hinton broth and chicken meat homogenate on stainless steel. And the second variant was to first prepare a preformed Pseudomonas fragi biofilm, which was incubated for 1, 3, or 7 days, followed by addition of Campylobacter jejuni for 24 hours. Everything was done with, lastly, with colony forming units to evaluate the results. Now, as you can see from the graph, and that was reported, was done with our GLM analysis, you can see that numbers for Campylobacter jejuni alone could not be determined after 3 or 7 days, but we could determine these numbers when Campylobacter was in combination with Pseudomonas fragi. You can see that numbers are lower at 5 degrees compared to 15, and at 5 degrees there was also a difference between what we obtained in chicken meat homogenate, where values were lower compared to broth. This quadratic trend at 15 degrees, we were able to correlate it with how Pseudomonas fragi biofilm biomass was formed, but these results are not shown on the graph. So our speculation here is that Campylobacter jejuni, when it's alone, it cannot adapt to the stress that's happening in aerobic conditions, and it transits into viable but non-cultivable state. But when in combination with Pseudomonas fragi, it gets this support from Pseudomonas, and it can retain its cultivable state. Now, interestingly, when we use this second scenario, where preformed Pseudomonas fragi biofilm was used, there was basically no difference between 5 and 15 degrees, and also Campylobacter would increase in number only when Pseudomonas biofilm would also increase in biomass. So we showed that Pseudomonas fragi improved cultivability of Campylobacter jejuni at low temperatures, so at 5 and 15 degrees. And it's also known that Pseudomonas can reduce oxygen tension. This was reported before in the literature. We also could measure this reduction by 38 to up to 59%. So our belief here was that this oxygen gradient is done in Pseudomonas biofilm that somehow improves stress response of Campylobacter in aerobic conditions. But as I said in the beginning, Campylobacter can be really moody when working with it in laboratories, so we couldn't perform these new additional experiments at low temperatures. So we had to do optimization step before proteomic analysis, and we chose 30 degrees in aerobic conditions for this analysis. Just short points. So we prepared plates for incubation. Proteins were isolated and purified. We did database searching, and at the end, quantitative proteomics with statistical analysis. We, all in all, I determined 149 proteins. 9 of them were downregulated and 19 up. From downregulated proteins, there were some for oxidative stress, acid stress, translation, and also in upregulated proteins, there was a bit more diversity, again, with oxidative stress, translation, phosphorylation, general stress response, transport of proteins. But as said in the beginning, we were more interested primarily in this oxidative stress response, which is quite unique regulatory mechanism in Campylobacter jejuni, which is considered from 5 regulators, and then antioxidant enzymes that on one side present superoxide defense, and on the other side peroxide. And also we have proteins for purification and protection of damage in DNA. And from our results, we could see that superoxide dismutase B was the one that was upregulated, meaning that this first line of defense in this line was improved, and also BCP and TPX, which are the ones important for aerotolerance in Campylobacter, were upregulated. Additionally, the protein responsible for detoxification of oxidized methionine proteins was upregulated. But of course, these are not the only, we cannot look only at this oxidative stress response. There are also other proteins which were up and down regulated. I will not go through all the maybe chaos in this figure, but what's really stressing is that all this information lead to this, that in these conditions with Pseudomonas fragi, Campylobacter jejuni can maybe improve its stress tolerance. It can also lead to increased virulence and increased antibiotic resistance. But there's still some work for us to do here to really confirm this information. So we show that Pseudomonas fragi reduces oxygen tension and improves Campylobacter jejuni cultivability at low temperatures. This upregulation in superoxide defense is this first line of fight against toxic byproducts from oxygen reduction cycle, and it's probably the one that's better to prevent additional steps in this reduction. This adaptation, as I said, can lead to increase of virulence and antibiotic resistance. So all this information indicate for us that this joint growth could increase the possibility of Campylobacter jejuni transmission to humans. And we believe that these results can also be applicable to other Pseudomonas that can be found in chicken meat or cow's milk or maybe even water, and also to other bacteria which can adapt well to these environments where we can find Campylobacter and provide it with protection. And there's, of course, really big question about what's happening with horizontal gene transfer, because Campylobacter is naturally competent bacteria. So my question, which I ask all of you, is if Pseudomonas fragi or other spoilage Pseudomonas are really just this quality problem, and if we talk enough about these bacteria, which on one side, of course, cause really big economic issues, but on the other side, we think that they have really big influence on pathogenic bacteria. So please see also more of our group's work on posters. And I would like to thank the organizers for this opportunity to talk about our work. Our group, Food Micro at Biotechnical Faculty, Dr. Marjorie Fournier, who helped us with proteomics, and of course our Slovenian Research Agency for financing. And thank you.
Thank you, Meta, for this clear explanation. Do you have any questions? Yes? Here?
Some Pseudomonas fragi strains are known to produce siderophores that can help iron transport. And sometimes the side error first, it's not only for Pseudomonas. They can give their side error first. So do you think the mechanism you saw can be a consequence of this?
Yeah, we saw that it influences also iron metabolism, but we have to do some additional bioinformatics to see about it. Yes.
Is there any questions?
I have one question. Are you planning to investigate, maybe you said but I didn't catch, other mixed biofilm with Campylobacter and others like Listeria or...
You mean other bacteria?
Yeah, to study interaction with Campylobacter, this kind of biofilm pairwise interaction with Campylobacter with some others.
We know that similar happens with pathogens like E. coli and Listeria. When you put them together with Campylobacter, we know that they, on electronic pictures from electronic microscope, you can actually see that Campylobacter is in the middle and the other bacteria go around, or maybe Campylobacter is the one that goes inside. And also with Bacillus, also from our group research, we know that it can support Campylobacter. So I think that, as I said, bacteria that can adapt well to such environments that we can find Campylobacter in, that Campylobacter take advantage of this and kind of hides in these biofilms. With also chemotaxis was activated, so yeah.
Okay, thank you.
Okay, thank you for all. So we can acknowledge all the speakers of the session and also all the participants for their questions. So I think it's a last talk and we can... Pascal has something to...
If by chance somebody found a cellular form, it may be mine, so just you can put it at the registration desk, or if it's in here, you can give it to me. So, thank you. Sorry for this.
So have a nice evening and see you tomorrow morning. Bye.